Taste and smell are both the perception of chemicals in the air or in our food. They are separate senses with their own unique receptor organs, yet they are closely related. Have you ever pinched your nose while eating food you dislike? Anyone who has done so before can attest to the difference in the flavor of a particular food when the sense of smell is impaired. This is because the flavor of the food is perceived through a combination of taste and smell. When our nose is pinched, we are only able to detect the taste of food and are unable to detect the food odors, therefore altering the flavor of the food. How do we taste food? Tasting food is essentially distinguishing food chemicals we are able to detect. We have 5 main tastes: sweet, sour, salty, bitter, and umami, which is savory. And those flavors are detected by our taste buds. Taste buds are specialized receptors widely scattered throughout the oral cavity. Most of your taste buds are on the tongue, but they are also scattered across your cheeks, your soft palate, your pharynx, and your epiglottis. So your taste buds are formed around structures called papillae, and there are 3 types of papillae on your tongue. There's the fungiform papillae, the foliar papillae, and the circumvallate papillae. The fungiform papillae are focused in the anterior region of your tongue, while the foliar papillae are on the sides of your tongue, and the circumvallate papillae are towards the posterior region of your tongue. So, most taste buds are packed deep down between the fungiform papillae of your tongue. A fun fact is, fungiform papillae are what gives the tongue its rough texture. So if you drag your fingers across your tongue, you can actually feel the fungiform papillae. Pretty cool, right? So this is a taste bud. And your taste buds are in between your stratified squamous epithelial cells, and every person has between 5,000 to 10,000 taste buds. Each taste bud has 50 to 100 taste receptor cells, which are stimulated by tastins, which are the food chemicals. And every individual receptor cell on the taste bud is activated by sweet, sour, bitter, salty, or umami. So taste ports here are essentially openings on your stratified squamous epithelium. And here's the gustatory hair. So here we have the gustatory cells in the blue. These are the cells that actually do the tasting. And we have the basal cells in the green. So, have you ever burned your tongue while eating hot pizza or drinking scalding hot coffee? Ever wonder why your tongue recovers in just a few days? Well, that's because the basal cells actually replace the gustatory cells every week or so. So the basal cells are the stem cells of the taste bud. So how does this all come together? When your saliva breaks down food into tastins, here are the tastins. Some of them will diffuse through the taste pore and bind onto the gustatory cell. Here's the taste pore, and they will bind onto the gustatory cell. And when the gustatory cells are stimulated, they cause signals to be transferred to the ends of the nerve fibers, which send impulses along cranial nerves to the taste regions in the brainstem. And from there, the impulses move to the gustatory cortex of our brain. Fun fact, there's a hypothesis called the labeled line hypothesis, which states that each taste receptor cell has its own dedicated labeled line, which means each taste receptor has its own dedicated axon all the way to the gustatory cortex of the brain. Here's a question for you. What happens when you are no longer able to detect sweet and salty tastes? Will you lose weight because you will no longer enjoy food? Will you become depressed? And how much will this really change your life? So, let's move on to smell. How do we smell food? Well, since the nose is our primary organ for detecting smells, the process begins with sniffing mold up our nose. Chemical particles called odorants and are filtered by your nose hairs here. And this is your nasal cavity here. So the odorants that do make it all the way through to the back of your nose and hit your olfactory epithelium here bind to receptors on your olfactory sensory neurons, which are these. Okay, so let's learn some of the structures involved in the olfaction process before I can tell you how it all works together. First, we have the olfactory sensory neurons. And that's not just the red, there's the yellow, green, blue, and purple. They're all olfactory sensory neurons. And there are columnar supporting cells in the olfactory epithelium. You can see here the columnar shape. And there's the cribriform plate, which is this bone here that separates the olfactory epithelium from the brain. And there's the olfactory bulb, which is actually in the brain. And there's the glomerulus, which the olfactory sensory neurons connect to. We'll go into more detail about that later. And the mitral cells. So, odorant activates an olfactory sensory neuron. So an odorant, once it binds and activates an olfactory sensory neuron, it'll send an action potential through the cribriform plate here, the cribriform plate, to the glomerulus in the olfactory bulb of the brain. And here's the glomerulus. And then the glomerulus synapses to the mitral cells, which will then send signal to the olfactory cortex of the brain. And just like with taste cells, olfactory sensory cells are only sensitive to a single molecule. The olfactory sensory cells are sensitive to the same molecule, will actually bind to the same glomerulus. So all these bind to the same glomerulus, and these in the rainbow colored here bind to the same glomerulus as well. So why does this happen? Well, our body is an extremely efficient machine, and the process via glomerulus is simply much more efficient than every single neuron, every single olfactory neuron sending their individual action potentials to the brain. So you can think of it like voting. We vote and we elect MPs, which represent what we say. And in the case here, olfactory sensory neurons are kind of like us, and the glomerulus are like the MPs. So the olfactory sensory neurons detect the same molecules or votes, and then for the same glomerulus. So question 2. What are the similarities and differences between the glomerulus and the taste bud? Thank you for watching.